We are Group 17, and our project is over genetic testing for breast cancer. Breast cancer is a cancer that primarily occurs in women and forms in the cells of breast tissue. Only about 5 to 10% of breast cancer cases have a genetic cause, with the rest being due to environmental factors. It is important to note that there is a 2% base risk for developing breast cancer, so any increased risk we present is relative to this base risk. In general, the casual mutations that are recognized to significantly increase breast cancer risk within families are quite rare. These include mutations in the following 22 genes. However, the primary genes considered to have risk alleles include BRCA1, BRCA2, ATM, CHEK2, and TP53. These genes are all tumor suppressors, which means that each is responsible for producing a protein that monitors the cell cycle and tells the cell when to stop reproducing. When there are mutations in these genes, the proteins do not work effectively, and the cancer cells are able to rapidly divide without any regulation. Genes that have strong predictive values for breast cancer and genes that we will be focusing on for our SNP chip test are BRCA1 and ATM. These genes have SNPs that independently present a low increased risk. However, the combined result can be a much higher risk. Therefore, these are not the only two genes with a strong predictive value for developing breast cancer. BRCA1 is a tumor suppressor gene that codes for proteins that repair DNA located on chromosome 17 and has an autosomal dominant pattern of inheritance. Sometimes, one nucleotide in the DNA sequence is replaced with another, which changes which amino acid is coded, which can completely alter the structure of the protein and, as a result, the function. While there are many possible variations, one example with a relatively large increased risk is SNP RS1799950. The G allele is the risk allele in these variations. The AG genotype is linked to an increased risk of about 1.5, and the GG genotype is linked to an increased risk of about 2 times. ATM, located on chromosome 11, is also a tumor suppressor gene that codes for a protein located in the nucleus of the cell, which regulates the rate at which the cells grow and divide, and has an autosomal dominant pattern of inheritance. SNP RS1800056 changes the two different versions of this SNP are C and T. The CC genotype is linked to an increased risk of developing breast cancer of about 1.5 times. And the genotype of CT is linked to an increased risk of about 1.05 times, making the C the risk allele. The information provided by our SNP chip test will help determine whether or not a patient has a predisposition for developing breast cancer. If the patient has one of the genotypes we have previously mentioned, then the patient will have an increased associated risk as seen on this slide. Because these percentages are so low, our test cannot tell a patient for certain whether or not they will develop breast cancer in their lifetime. After receiving genetic testing results, an individual must decide what to do with these results. If the test comes back positive, meaning that you have a mutation in one of the genes that is linked to an increased risk of developing breast cancer, it can be very difficult for both the individual and the individual's family. The individual could choose to act preemptively and get a mastectomy. However, there is still a significant chance that they never develop breast cancer. Not to mention, these surgeries are incredibly expensive and could potentially be a waste of money. While many people are hesitant about getting genetic testing, it is almost always a good idea. It is better to know and to have the choice to act accordingly than to not know until it's too late.